Welcome back to the Back Bay in Boston for a live broadcast now of the first day of the World Medical Innovation Forum. Our next panel is focused on an area of intense interest, CAR T, its status, lessons learned, and what's next. And we have one of the leading experts in the field to moderate, Dr. Marcella Moss, who we work very closely with. Delighted to turn it over to Marcella to lead this session.
Thank you so much, Chris. I'm thrilled to introduce a wonderful panel today. We're going to go through some questions. I have with me Jake DuPont from Atara Biotherapeutics, Kristen Heggie from BMS, Stefan Hendricks from Novartis, and Christy Shaw, who's the CEO of Kite. So I'll just call on panelists by name, and we have some questions for all and some that I'll ask a little bit more targeted. So let's get started. So the first question is CAR T still an industry priority? Are new investments being made by the large companies, and what kind of interest level are you getting from the small company perspective? So Jacob, why don't we start with you?
Absolutely. So thanks, Marcella. And full disclosure, I'm speaking from the small company perspective. I'm at Atara Biotherapeutics, and I will say that there really is, in my view, a lot of interest in CAR T therapy. I think this is the dawning of the age of this type of therapy. So we've seen the 5 approvals that have come through for auto CAR T, and this is really the crest of the wave. There's so much innovation that can come after that, whether it's innovation of new armoring technologies, new co-stimulatory domain, allogeneic approaches, maybe moving from liquid tumor into solid tumor. I think there's so much opportunity for innovation that it is certainly a very hot time for CAR T therapy. I will also say from the small company perspective, we actually see quite a lot of interest from large pharma in terms of the work that we're doing. And we actually, to this point, established a big pharma partnership at Atara with Bayer in December of last year because of their interest in allogeneic CAR T cell therapy. So I think that's case in point that there really is a lot of interest in CAR T.
That's great. Kristen, can you tell us your perspective from BMS?
Sure. I think I'd concur with others on this panel that CAR T and cell therapy in general is a huge priority for the industry, big and small pharma. I mean, I personally have had the opportunity to live through the partnership at Celgene with Bluebird, the acquisition of Juno, and then the BMS acquisition of Celgene. Just sort of emphasizing the relevance of cell therapy. And at BMS, of course, we're partnering with small companies in the cell therapy space all the time. We have 4 active partnerships in the area. And Christy, you mentioned our last panel, I think, was some huge number of new companies starting in the cell therapy space. So, there's an innovation engine. There's, small and big biopharma interested in the space. It's just a very exciting time to be in cell therapy.
Yeah. Christy, do you want to tell us more about it?
Sure. And I agree. This is, I think, continues to be a hot area. It's not easy or for the faint of heart or for the people who have small money in the bank, because obviously it's a very time intensive and cost intensive, but it's really the only area in oncology, or one of the only areas, where we can talk about a cure. And, you look at patients who are given 3 to 6 months to live in DLBCL, for example, that are now, specifically on Yescarta, they're 4 years out, 44% of them are alive. And it's just remarkable. So I think that gives everybody hope to continue with that. And, we have indications, as Jacob talked about, 5 of them that we have, and the response rates are just tremendous. So it's worth the effort for sure, even though it's not easy. And one of the things we talk about too is, when we say it's not easy, the manufacturing is so important. I'm not sure everybody always gets the connection because, manufacturing in small molecules is, inventory on the shelf. Manufacturing in cell therapy are cells that are patient. and they need them back, and there isn't anything on the shelf. And so that makes it critical on not only the manufacturing, but that supply chain. I'll give you an example right now in Israel with the crisis there. We have 4 patients where we're trying to figure out how do we get their apheresis? How do we actually get back in the country? And so we have people who are used to getting medical supplies in war-torn countries helping with the supply chain. So it's that kind of intensive work from end to end. So, but I will say, as Kristen was talking about, that there are about 120 companies, and they're all working really hard on the science. And what we're trying to do is partner with them so that we can figure out how to help them commercialize it when it becomes time. And so I think it is a great partnership.
That's great. Yeah, we'll come back to manufacturing a little bit in the next couple questions, but I wanted to give Stefan Hendricks from Novartis a chance to say, is CAR T still a priority at Novartis?
Well, absolutely. The short answer is yes. And, we at Novartis, we truly believe that cell and gene therapies will play a major role how we're going to treat human diseases in the future. So that's why we are very committed for the long term and learning and improving. And as said, other big players are making investments to join this field. Hundreds of smaller players are working hard to enter the field. And also on our side, we continue to invest in strengthening our capabilities, developing next generation technologies and investing in a broader pipeline. So it's super exciting space. And on a personal note, I'm in this biopharmaceutical field for more than 25 years, done multiple therapeutic areas, but working in this field of cell and gene therapies, getting so close to patients and indeed, as Christy said, sometimes dealing with amazing challenges in countries, and then seeing what the team is able to do to help these patients, and then the promise of a cure for those patients, it's really inspiring to be active in this field.
I couldn't agree more. In fact, I'll just make a little side note that of the people that I've worked with, either both in industry and academia, once you work on cell and gene therapy, it's really hard to go back and work on anything else. So I think that's generally good for our field as we recruit people into it. What do you think that we've learned from first generation products and the efforts to bring these to the commercial setting, especially with regard to the manufacturing challenges, some of which Christy already alluded to, but I wanted to start with Jacob for this question.
Yeah, thank you, Marcella. So, I think, and people have already made reference to it, I think what we learned with the first generation products is that these really work. These make a huge difference for patients. So, we're using the cure word, at the podium here, and I think that is incredibly meaningful. And these products are administered to patients who have received other therapies that have failed them. So this is a big deal that we've learned from those first products. And I think it's also this aspect of manufacturing, and as Christy was also referring to, we are figuring out how do we produce these cells. They're obviously, the first generation are all autologous. So how do we make these for patients? A huge logistical issues, as Christy was referring to as well. But we're getting better and better at it. I think we've also taught, if you will, and worked with regulators where, regulators have figured out how to approve these drugs, which is really meaningful. And so that makes a big difference. I think we've also learned a lot about what does the cell product need to look like? I think it's really interesting. You talk about the phenotype of the cells, the number of cells that you have to give, the preconditioning regimens that you have to think about. What are some of the other aspects that you have to deal with? Antigen loss, tumor burden, all those types of things. And that's just for heme malignancy. So I think we've taken the first step. We've gotten the first approvals that are making a huge benefit. But the world is our oyster in many ways when you think about what are we going to do next? What are the next binders we're going to go after? What is the second or third generation co-stimulatory domains that we're going to use? What about armoring? What about going into solid tumor? I think it's really incredibly exciting. And frankly, another area that I'm really interested in is what about allogeneic? What about off the shelf? What about, getting these products to patients based on an inventory where they can get treated, 2-3 days later after they get identified. I think that's incredibly exciting.
Yeah. Christy, do you want to maybe build up on some of what you mentioned before about what we've learned about in terms of manufacturing from this first generation of products?
Sure. The manufacturing, like I said before, is key right now to get to patients in the community. We see actually only 4 out of 10 patients are being referred for CAR T that are all eligible. That means 6 out of 10 patients that are eligible are going to palliative care. And so the biggest thing we need to do is make sure we get more patients the ability to access the product. And that consistency of manufacturing is important in that. And I agree with Jacob as well that, ideally what we want to do is take what we've known, these literally hundreds of steps between, initiation and final distribution back to the patients, literally hundreds, that we need to look at how do we automate that. So from an autologous standpoint, how do we build the machinery that can do what humans do, that can do it faster? So far we have in the US one of the fastest turnarounds at 16 to 17 days, but we need to be even faster because also, as Jacob said, these patients this is their last resort, and a lot of them don't have time. But then also as we move to the future, it's not ready for prime time yet, but we hope it will be in the not too distant future to be able to have off-the-shelf allo, and we'd love to participate in that. And we have some of our own programs, but that would be ideal, as if we could just take it off the shelf and give it to these patients and have them not wait. So I think I agree with all of that, and being able to manufacture successfully and be able to take that manufacturing and reduce the cost and increase the speed.
Yeah, that would be a transformative difference. Stephanie, can you tell us a little bit about Novartis and what you've learned in terms of first generation products and in the commercial setting with an autologous product? You guys have been in the clinic now for a couple of years, so.
Yeah, the journey started 9 years ago with the collaboration with UPenn University, and so a lot of learning since then. And when you go first, there's no playbook of how to introduce a one-time treatment with potential curative attributes, right? So I think very importantly, one of the focus points was to how we want to get this fantastic option to a lot of patients worldwide. So the first thing that you have to do is create access. And also for our payers, this was a very new field. So the first thing we had to do is to really understand their needs, and they were different, right? The way value is being defined can be very different from country to country. So learning how they define value, calibrate that, learning their needs, and then partner and come up with innovative solutions to agree on pricing, reimbursement, and then make it available for patients. And I think we learned a lot, and we have implemented many innovative solutions, and we are now reimbursing 28 countries worldwide and opening more countries this year. So I think a lot of learnings there. And then the other thing, as already alluded to, is manufacturing. And I think on that front, I think on the CMC front, it is so important that you have a continuous dialogue with the regulators, because they were learning too. So they actually learn together. And I think we have made great progress. I mean, actually right now we have 6 commercial sites available worldwide, being able to meet global demand and being closer to patients. So that was great collaboration with the regulators. And also through the years that we're now in the market, working with regulators to further improve the robustness of the process step by step, and through multiple steps, we have now a very high level of robustness, making sure that when patients, give us their cells, they trust us with their cells, that we're able to return a high quality product. So I think a lot of learnings there. Maybe the most important learning is that we've learned it's so important to learn together with all the stakeholders and partners out there. It's a great journey, and I think Jacob said it, we're only at the beginning.
Yeah. So maybe like some of the other learnings are, we've mentioned, we've all said the word cure, which is sort of impressive for a group of oncology-oriented developers and oncologists. So maybe just keeping it on Stefan for a moment, can you tell us a little bit about what we've observed so far with the Novartis products, about how enduring these responses are, and what are the most common forms of relapse have been?
Yeah, happy to start. I think what is great news for everybody working in this field is that with a lot of new data emerging, we see more and more confirmation of the durable promise of these therapies. And I will highlight a few. As I already mentioned Japan, they very recently published in the New England Journal of Medicine, they published 5 years data. So that's the longest follow-up available in the market. And I think a great observation in their data was that the patients that responded, that were still responded at 1 year, that was maintained up to 5 years. So I think that is a great message on durability. Also, we have done our own follow-up data up to 40 months with the Juliet data. And also there we saw that the responders, the majority of the responders, they maintained their response up to 24 and 36 months. So this is another confirmation. And I think what's really important is that we have now seen around the world multiple real-world evidence data sets that confirm similar efficacy numbers in the real world that what we have seen in randomized clinical trials. And I think so everybody can become more and more confident in the durability. Now, on your other question on reasons for relapse of not responding, in that field, there's still a lot to learn. So there's still a lot of unknowns and there's still a lot to learn. But there's a few things that we know and that we can act on and everybody can act on these days. I think what is important is what is the lymphodepleting chemo that you choose. If you use Flucci, you know that you have better response rates, right? And very important, maybe the most important is that we see patients coming in earlier. Unfortunately, around the world, we still see patients coming in after 5, 6, 7 rounds of chemo, even up to 8 rounds of chemo. And if these patients can be referred earlier and treated earlier according to the labels of these fantastic medicines, I think these patients have a much higher chance of success. So there are things that we can do today, and there's things that we still have to learn.
Thank you. Christy, just sticking to the CD19 theme before we move on to myeloma, what can you tell us about durability and mechanisms of relapse with the axi-cell products?
So taking off what Stefan said, we're really excited to hopefully move into earlier lines of therapy to be able to help more patients. We'll see some of that data coming out, and our studies on track for submission this year. But I think that's when patient cells are healthier, when they have more time, is when, also would relieve some of the pressure in the supply chain and manufacturing. So we'll see what that data shows. And it would be, to Stefan's point, this could be industry changing. On the response rate for Yescarta, it's one of our biggest issues as we look, what are we trying to solve for in the future? One is, how do we make these differently, like off the shelf? But even more important than that is why are almost half the patients have durability up to 4 years. How are we going to help the other half of the patients? What is wrong? Why are they not responding? So we're doing a lot of translational work with academic centers and with our own samples to try to identify those patients and look at things like bisystronics, look at things, different types of therapies, et cetera. So I think that next piece of really trying to use combination therapies is going to be really important for us as well. But I want to balance too, that these are very toxic therapies, and that, whether it's, neurotoxicities or CRS, that's also something as we look at the risk benefit that, if we can reduce those and increase the efficacy, that will continue to make the autologous feel sustainable as we move to off the shelf eventually.
Yeah. Kristen, do you want to tell us about the experience in multiple myeloma in terms of how enduring the responses are and how they're similar or different than CD19?
Sure. Yeah, no, well, so we're very excited. It's been quite a year at BMS with 2 back-to-back CAR T-cell approvals, one for CD19 and one for BCMA, Breyanzi and Abecma. With BCMA, this is a first-in-class CAR T-cell therapy for multiple myeloma. And we have been able to benefit from, now data in more than 1,500 patients across, both of our products, across 6 different disease indications to try to get at some of these questions. What drives response? What drives early relapse? What drives late relapse? I think it is clear in multiple myeloma that the shape of those relapse or progression-free survival curves are different than they are in DLBCL, which is not surprising because historically myeloma has been an incurable disease, whereas historically DLBCL has been cured with chemotherapy. It's been cured with autologous stem cell transplant. So, we're diving now into what are those mechanisms of relapse after CAR T cells in multiple myeloma. And what we've learned is more what it's not. We have not seen a high frequency of BCMA antigen loss as a mechanism of escape. We have reported a couple of case reports published this year, in a couple of patients, about 4% of the population, we have detected, bilateral BCMA antigen loss in general in patients who are coming in with one allele, either deleted or mutated, and then they acquire a mutation or deletion in the second allele. But that is not the dominant mechanism of relapse. And we're now, like others, using these deep translational databases to understand the patient factors, the product factors that will predict either durable response or early progression. Some of the observations we've made are relatively simple. Like we've shown if you clear your soluble BCMA at month 2, you can dramatically differentiate patients with early progression versus those with very durable response. And at least that gives you an early post-treatment look to separate out patients who are likely to have a durable benefit. And for the others, we can consider, other strategies, combination therapies, et cetera, to get deeper responses in those patients.
Thank you. So since we're running short on time, I think I'll ask the last question of all the panelists here. What would you say comes next for you in terms, or for your company in terms of beyond CD19 and BCMA? What is your major priority for how CAR T should move forward? I'll start with Kristen, sorry.
Okay, well, I'll keep going then. Yeah. We are very interested in looking at new targets beyond BCMA and CD19, both in blood cancers as well as in solid tumors. For solid tumors, obviously pivoting somewhat from the CAR T cell approach to the engineered TCR receptor approach. And we have 4 active partnerships, many of which are looking at novel solid tumor targets for TCR approaches. And as others have mentioned, looking at dual targeting approaches, arming your cells with novel receptors to tackle the immune inhibitory microenvironment, off-the-shelf CAR T cells, either derived from donor-derived cells or even from induced pluripotent stem cells. So we're very active and engaged in the whole space. We're interested in cells beyond T cells, NK cells, gamma delta T cells. So I think the next 5 years are going to be very exciting.
I agree. Sounds like a lot of priorities. How about Stefan?
Well, I think when I think about the future, I think about 2 things, right? I think about how can we reach more patients and how can we improve the outcomes for those patients. And when I think about more patients, I think about next generation platforms and next generation technologies that will make it easier to reach more patients. And we're investing heavily in that. Then the other thing is, as also Kristen already alluded to, is looking at more targets and finding ways to bring these therapies to more diseases or like more indications and more malignancies. So I think that is about more patients. And then when it is about more better outcomes, I think I look at it from twofold. One is we have new technologies like CRISPR and other technologies that we can modulate the cells and further improve the cells and improve durability, improve, all kinds of edits that will help us get better outcomes in the future. And the other part is building better constructs, right? Building duals or other ways of further improving the constructs. And I think that's how we focus.
Wonderful. So, Christy, 30 seconds. What's your next highest priority?
Sure. So, in the last 18 months, it's really been busy. Even during the pandemic, we received approval for MCL, follicular lymphoma. We're able to do by video or Zoom the manufacturing inspections in Europe with FDA Watch. And so it really is moving forward quickly, as Chris and I have talked about before. And regulatory authorities are really partnering well with us. We're still looking at this year at an approval in CLL. We also are looking next year at adult ALL and hopefully the second line approval. And beyond that, our bicistronic is looking to move to IND this year. But most of what we're seeing, I mean, there's so much more to do in the blood cancers. We have not treated all of those patients. So we're really focused on that. But we're also looking at a partnering strategy where we let these small companies that are really focused on their therapy and their platform to help them and partner with them to bring those to fruition. So it's one of the strategies of trying to get this external innovation going even faster so we can help more patients.
Fantastic. Jacob?
Yeah, absolutely. So I think for us, it's really the focus on allogeneic cell therapy, and that comes in three major priorities for us. Our platform is allogeneic EBV specific T cells. So we're in the process of preparing our BLA submission for tab-cel to get the first allogeneic T cell regulatory approval, and we're guiding towards that BLA submission here. So that's foundational for us and get that first one approved. Then the second is our MS program, which we think is really transformative potentially. So that's allogeneic EBV specific T cells for progressive MS. We're going to have phase 2 data at the beginning of next year. And then in terms of CAR T, which I know is the focus of this forum, our allogeneic EBV T cell platform here, where we think these EBV T cells really could be the secret sauce, if you will, or a great foundation for allogeneic CAR T, where you can then transduce with your scFv against CD19 or solid tumor targets. You can innovate with the next generation co-stimulatory domains like 1XX or Michel satellite, and then you can armor with things like PD-1 dominant negative receptor, which we think could be important for solid tumors as well. So we're really focused on allogeneic T cell therapy, and our platform are these EBV specific T cells.
Sounds great. I'm glad to hear you all have the same priorities that I do, and I'm hoping that as a field, we really take it to the next level. So sounds like it's going to be an exciting ride.